Good morning, everyone. My name is Shoji Saito from Matsumoto City, Japan. I'm a pediatrician, mostly working on hematology and oncology. And first of all, I deeply thank organizing committee to give me such nice opportunity to present our work. I'm really happy to be here in such wonderful conference in a beautiful country. Yeah. So both me and Shigeki Sensei are coming from Matsumoto City, Japan. I guess many of you may not be very much familiar with our city, so just a brief introduction. So our city is a kind of small town surrounded by Japan Alps. But we have lots of tourist spots, including historical castle or very scenic place. So it takes about 2 to 3 hours from Tokyo or Nagoya. So I hope you guys come and visit Matsumoto City in very near future. We Sensei know very well about that. Yeah. So here's my talk. So let me first briefly overview the CAR-T therapy. So as Professor Patsai already showed you the function, but so basically chimeric antigen receptor CAR is a synthetic receptor that combines antigen recognition domain and signaling domains. And the structure of antigen recognition domain generally are derived from scFv region of antibody. And most important point of CAR is that theoretically you can target any cancer antigen by replacing anti-binding site, and you can modify CAR structure whatever you want. And many of you may also know that the CAR-T cell therapy showed great success, especially against B-cell malignancies and multiple myeloma. And several CAR-T products have been approved in the US, Europe, and also in Japan. However, there still remains challenges regarding CAR-T therapy, especially for limited efficacy outside B-cell malignancies and multiple myeloma. And safety is also a concern because even in CD19 CAR-T cells therapy, so we sometimes observe very severe adverse events like cytokine release syndrome or neurotoxicity. And when you want to develop new types of CAR, you really consider the on-target of tumor effects because your target, if your target are expressed in normal cells, maybe your new CAR may cause severe adverse events. And also, as Professor Patai already showed you, that the cost is also the problematic because we needed to manufacture GMP grade CAR-T product individually, and most and all clinically available CAR-T products use viral vector to introduce CAR gene into lymphocytes. So it also costs you additional expensive money. So to overcome these issues, we are trying to develop an alternative method to improve the efficacy while reducing the cost of CAR-T therapy. So let me move on to the next session. So we have been working on piggyBac-CAR-T system for more than 10 years. So our boss, Professor Nakazawa, developed this system when he was in CADT, Bella College of Medicine. So I won't go into details, but basically this system uses piggyBac transposon and transposase from cabbage rooper to introduce CAR into T cells. We use electroporation to integrate CAR gene into lymphocyte genome. And the most advantage of this system is that this system only uses DNA plasmids and electroporator, but not requires any viral vectors, which can reduce the cost of CAR-T manufacturing. So this is a quick summary of the characteristic of piggyBac CAR-T therapy. So although transfection efficiency is much lower in PB than viral vectors, we believe that this system has several advantages, such as less immunogenic and no need for T-cell activation before transfection, so that probably preventing T-cell exhaustion. And most importantly, we may be able to reduce the cost a lot because we don't need viral vectors. Okay, so let's move on to the next slide. So to apply this system to treat hematological malignancies, so we are developing several CAR T cells using this system. And my topics is mostly working on AML, but let me just start with CD19 CAR T cells. So at the beginning, so we are trying to apply this system to CD19 CAR T settings. And as Professor Shigeki Sensei showed you yesterday, so we are struggling to improve the manufacturing efficiency. And mostly we are working on modifying manufacturing protocol, not piggyBac itself. And somehow we managed to get better efficiency, as you can see here. So by using this method, so we got good efficiency and potent anti-tumor effects of piggyBac CD19 CAR T cells. And we next try to apply this system for treatment of AML, acute myeloid leukemia. So as you may know, so AML is the most common leukemia in adults and the second most frequent one in children. And the prognosis remains poor, especially in those who relapsed or refractory. So novel treatment strategies are urgently needed for AML. And yeah, of course, many researchers have been working to develop a novel CAR-T therapy for AML. But unfortunately, no commercially available products are available, even so far. To develop novel AML targeted CAR-T therapy, so we specifically interested in GM-CSF receptor, GMR. So GMR is a unique receptor for GM-CSF, consisting of CD116, GMR alpha chain, and CD131 common beta subunit. And importantly, normal resting cells express this kind of GMR. So GMR alpha subunit alone, which has very low affinity to GM-CSF. But activated cells, including AML cells, express highly ordered GM-CSF, a complex receptor, which has very high affinity to GM-CSF. So when we designed our GMR card, we used GM-CSF itself as an anti-binding domain instead of using SCFB for GMR. In mild malignancies, AML generally express high levels of CD116, especially FAB subtypes M4 and M5, so myelomonocytic leukemia. And as we expected, so we also observed high levels of CD116 in both JMML and CMML. In contrast, we observed minimal expression of CD116 in ALL, acute lymphoblastic leukemia. And at the beginning, so again, so I think that the pediatric hematology oncology people know well that hypersensitivity to GM-CSF is a hallmark of JMML, juvenile monocytic leukemia, children's leukemia. So again, Professor Nakazawa has initially developed ligand-based CAR T cells against JMML. And yeah, indeed, GML CAR T cells showed potent anti-tumor effects against JMML. But interestingly, GML CAR T cells didn't cause such cytotoxicity on normal hematopoietic stem cell. And further improve these GML CAR T cells, we try to modify GML CAR structure. So this is kind of back to the basic slide. So when you look at the CAR molecules, there are many parts to be modified, but we first look at the spacer region. So spacer region is a tiny structure that connects antigen-binding site and the transmembrane domain. But many researchers showed that the length and flexibility affect much for the function of CAR T cells. So this is one example of the spacer really affects the function of CAR T cells. So this is Dr. Watanabe, CADT, have done this work. So he extensively compared a variety of CAR with different types of spacers. And he shows that CAR T cells with different spacer work differently. And he found that the intermediate length of spacer works the best in terms of tumor control, mass survival. And he also looked at the phenotype of T cells. And yeah, it also affects the T cell phenotype by spacer, by the types of spacer. So in our GML CAR T cells, we indeed optimize the spacer region. And actually, original GML CAR structure used CH2-CH3 region, which is known to abrogate the in vivo CAR T cell function. So we really need to get rid of this region. So to modify this region, so we basically tested two types of spacers, simply removing this region or just replace it by G4S spacer. And this is what happens. So it really makes a big difference. So as you can see here, G4S GML CAR T cells show the superior short time killing, this one, ability over delta CH2-CH3 spacer ones. And moreover, GML CAR T cells with G4S spacers showed better tumor control in THP1 AML xenograft model. So the spacer modification really improved the efficacy of CAR T cells. And we next wanted to modify antibody binding domain of our GMR CAR T cells. So this is a SCFB CAR T cell. So, but we are using ligand-based CAR T cells. So we use our CAR contains GM-CSF in this part. So, and this is also the example of antibody antigen binding site really matters. So this is very interesting paper showing that CD19 CAR T cells with lower affinity SCFV called CAT showed better anti-tumor control in both in vitro and in vivo. And they also showed a striking clinical efficacy of those low affinity CD19 CAR T cells in their clinical trial. So to modify our GMR CAR T cells, so we also try to modify, antibody antigen binding domain. So, and this paper is kind of old paper, but very important for us. So this paper shows that some GM-CSF mutants function differently as compared with wild type GM-CSF. So specifically, this paper shows that E20R and E20K mutants showed mutant lacks agonistic effect to induce cells proliferation, which may be better for a binder of CAR. And also this paper shows that those mutants have lower affinity to GM-CSF receptor to the GMR. So which may be better for us. So we utilize these two GM-CSF mutants as a binder of CAR. And as you can see in the right, so we observed the better killing effects of both E20R and the E20K muted GMR CAR T cells showed in short time killing at a challenging ET ratio of 1 to 100. And we also observed the better anti-tumor control of CAR T cells with mutated GM-CSF as an antigen binding site. And finally, so we combined these two modifications and we successfully improved the efficacy of GMR CAR T cells with G4S spacer. And this time, so when we combined the G4S spacer, this specific E21K mutation showed the best tumor control in a xenograft model. So we used this structure for our clinical trial. And we also look at the safety of our GMR CAR T cells. So first we looked at the GMR expression in normal hematopoietic cells. And as you can see here, while T and B and NK cells lacks GMR expression, we saw high levels of CD160 expression in both monocyte and neutrophils. But in terms of expression levels, monocytes showed higher expression of CD160 than neutrophils. And interestingly, so we did not see the cytotoxic effect of GMR CAR T cells on neutrophils in this specific experiment condition. And we did see monocyte cytotoxicity on monocytes as you expected. And as you also expected, GMR CAR T cells didn't do anything on B cells and NK cells. And we also did not observe any fratricide in ex vivo manufacturing of GMR CAR T cells. And this is just to show that piggyBac modified GMR CAR T cells showed favorable T cell phenotype with lot of CD45RA positive, CCR7 positive population, as Professor Shigeki Sensei showed you yesterday. Yeah, and we also look at the exhaustion marker of GMR CAR T cells, and we didn't see PD-1 and LAG3 expression in our GMR CAR T cells, while we do see some levels of TIM3 expression in CAR T cells. So GMR CAR T cells exhibit substantial anti-tumor effects against AML, and modification improves the efficacy of these CAR T cells. And we also observed the acceptable safety profile of our GMR CAR T cells. So based on these preclinical data, so we are now conducting a phase 1-2 clinical trial of GMR CAR T therapy. So the primary endpoint of to evaluate the safety and adverse effects of GMR CAR T cells, and we also evaluate the efficacy, PK, and cytokine profiles as a second endpoint. And importantly, these pictures are how we are treating the first patient. So some of you know Miki Sensei is now taking out our GMR products from the box, and then we're gonna sort them, and so this is me and trying to infuse GMR CAR T cells to first patients. So this is a schema, just a schema of a study protocol. So we basically do leukapheresis from patient's blood and the manufacturing process. During this time, so the patient will receive salvage chemotherapy. And we also give them lymphodepleting chemotherapy consisting of fludarabine and cyclophosphamide, just a standard lymphodepleting chemotherapy. And after GMR CAR infusion, so we observed 4 weeks period to DL dose limiting the toxicity evaluation. And we required hematopoietic stem cells transplantation following CAR T cell therapy, mostly because of the safety concern from the authority. Because we cannot exclude the possibility of neurotoxicity of our GMR CAR T cells. Also, we did not observe so much cytotoxicity on neutrophils in our in vitro evaluation. And our hospital luckily has our own in-house CPC and QC lab, so we can manufacture it all in our hospital. So now, GMR CAR T clinical trial is ongoing, and in Shinshu University, so with the great help with Professor Shigeki Sensei, so we are running how to CAR T cell piggyBac how to CAR T cell therapy for bone and soft tissue sarcoma. And Shigeki Sensei also just initiated the EPHP for his own CAR T therapy for Ewing sarcoma and solid tumors. So this is my conclusion. So piggyBac system can be utilized for CAR T manufacturing with favorable T cell phenotype and potent anti-tumor activity. And we extensively optimized our GMR CAR T cells and successfully improved the efficacy of this card. And now we are conducting phase 1 clinical trials of piggyBac T cells for AML. So I hope we can share clinical data with you in near future. So I deeply thank our project team member, physicians, nurses, and the patient. And this work is supported by Japanese AMED. And I thank all of you for kind attention. Thank you very much.